Hello and good afternoon, friends. Welcome to CEC-UGC live lecture. Dear friends, as you know, this session of ours is all dedicated to the science subjects, and today we are going to talk on cancer biology. We are going to discuss different types of cancer, how could be they diagnosed, as well as what are their treatments. For this, we have again with us in our studios Dr. Amit Bhattacharya. He is Assistant Professor in Ramjas College, Delhi University. So first of all, I would like to welcome our guest, Dr. Amit Bhattacharya, in this particular lecture of ours. Welcome to the EduSet lecture, sir, and I believe and hope the students who might be unaware with the different concepts regarding the cancer biology would be able to learn from this very lecture.
Yeah, thank you, ma'am. Today I will be focusing on the cancer biology, what exactly a cancer is, how it is caused by various different radiations or carcinogenic substances, what are the molecular phenomena which are causing these cancers to occur in the various body parts, which are very essential and vital for the functioning of the body. Subsequently, I will go to the diagnosis, how these cancers can be detected in our body, how they have spread through our body, and at the end I will come to the treatment part, what are the various treatment methods which are being used to treat the cancers. Before starting, let me start up with the historical perspective of the cancer biology. Today, cancer has become one of the most dreadful diseases which is affecting about 14 million people throughout the world, of which 8.2 million people are dying because of cancer. The cancer biology has been defined by the WHO as cancer is a rapid creation of abnormal cells that grow beyond their usual boundaries and which can invade adjoining parts of the body and spread to other organs. And this process through which they are passing on to the other parts of the body is called as the metastatic process. And subsequently, the patient dies of the metastatic growth. The oldest description of the cancer biology goes back to 3000 BC, when the Egyptian medical books in which it has been written the symptoms of cancer biology. But at that time, the word cancer was not used, but the similar kind of symptoms have been found in those inscripts. Subsequently, in the year 460 to 370 BC, the father of medicine, the Greek physician Hippocrates, for the first time described the cancer, and he used the terms carcinomas, which said to have the ulcer-forming tumors. Subsequently, in the year 200 to 28 BC, the word cancer originated, which was given by the Roman physician Celsius. For the first time, he described the word cancer, which Latinly means crab. Another Greek physician named the Galen, in the year 200 AD, for the first time described the onco, which means the tumor growths. Subsequently, it was found that the cancer growth has been found through the ages. The WHO, in some of the recent reports in 2012, have said that there are about 8.2 million cases of cancer patients who are dying of cancer. Of these, 60% of the cases are found in Africa, Asia, Central and South America. Of these, 30% of the cases are treated if they are diagnosed at the very early stage or the level. The cancer has been spread throughout the world. Through the world, there are about 14 million cases, of which about 8.2 million people dies of cancer. In India, there are about 1 million cases, of which about 0.6 million people dies of cancer, of which majority of them are from the women, while the other part is from the men. So there is an equal ratio of men and women who are getting affected by this cancer. Throughout the world, there are various forms of cancer which have spread, and of these, the most common cancer-causing deaths are of the cancer which are happening to the lungs, which demands to about 1.59 million deaths. Liver cancer, which states to about 7,45,000 deaths. Stomach cancer, which tolls to about 7 lakh deaths. Colorectal cancer, which is about 6 lakh deaths. Breast cancer, which occurs in the woman, is of about 5,21,000 deaths, esophageal cancer which rebounds to about 4 lakh deaths. So over here I have shown a picture of a cancer of a liver stage. So if we can see, the liver has been transversely sectioned and we can see there are thick growths of the tumors in the liver cells. And on the right hand panel there is a picture which is showing the stained slide of these cells. So we can easily see that the tumor cells are in much more high density number than the normal cells, and we can easily say that the growth has become metastasis and it will now move to the next stage of the cancer. Let me show you some of the picture of the other cancer. Over here I have shown the picture of a lung cancer, which is many a times known as the Pfeifferoka cancer, in which we can see that in the lung there are discrete and active growth of the abnormal cells which have grown to form tumors over there. The next cancer is a stomach cancer, which we called as Petka cancer, in which we can easily see the stomach has got many of the abnormal growth and subsequently the patient develops the stomach cancer cases. Third is the colorectal cancer, in which the rectal part where the absorption and the other things takes place, the colon part, the cancer occurs at that particular part, and we can easily see from the diagram there is active growth of the cells in those particular parts. And finally the oral cancer, which occurs in the mouth and the oral cavity. We can see that there is active growth of the cells or the tumors which have grown in the mouth, in the buccal cavity, as well as some growth in the tongue also. So from these pictures we can easily make out that the cancer is occurring because of the abnormal uncontrolled growth and cell division of the cells which are designated to die after some time. In our body, each of the cell has got a lifespan. So after a particular lifespan, the cell has to die. Like for example, the RBCs. The RBCs in our body has got a lifespan of 120 days, after which it automatically dies off and the fresh RBCs replaces these old and dying RBCs. The process by which these RBCs dies off is called as the programmed cell death or apoptosis. So let's take up an overview of the tumor formation. The tumor formation generally takes place in 4 major steps. The first one is the mutation, the second one is the tumor growth, the third one is called as the intravasion, and the fourth one is the metastasis. Let's take up what exactly happens at each of these steps. In the first one, what happens that the mutations occurs, in which the cells which are lining the glands or the organs, they gets mutated by some kind of carcinogens or radiations. So as soon as they get mutated, the normal functionality of these cells get overed up, or the checkpoints get released, and now the cells start growing rapidly without any checkpoints or stoppage. So now the cell grows over at that particular point to develop the tumor cells, which we call it as the benign tumor. So the tumor growth occurs when the tumor cells start growing over a period of time, over a short period of time, into a number of its progeny. The third point occurs is the intravasion, where these tumor cells, they get separated out or get released out from their point of attachment and goes into the blood vessels, and through the blood vessels they go and invade some distant part of the body, and at that particular point we call that the patient is suffering from metastasis, where the tumor from one particular portion is going through the blood vessels or lymphatic vessels and getting transported to some other portions, and over at the new position these tumor cells start developing over there. Now let me take up a animation over here. We can see that the tumor cells have grown with the mutations. They start growing in a larger number, and subsequently what happens that these tumor cells break through the epidermal layer and they move into the blood or the lymph vessels, where they go through the blood vessels into some distinct part, and at this distinct part again the tumor cells divide and they grow, and again they move through these blood vessels so to other part. So at the last stages of the cancer, most of the patients suffer from the cancer in which the spread has been found throughout the body. Now in the cancer, one of the major phenomena of the cancer is the angiogenesis, apart from the metastasis. Angiogenesis means the formation of new blood vessels, because as soon as the new cells are being formed in our body, these cells requires a active supply of the nourishment and the energy and the oxygen supply. So there has to be fresh formation of the blood vessels through which these supply can be provided to these freshly formed tumor cells. So the angiogenesis process starts, in which from the primary tumor, as soon as it is formed, new blood vessels start forming and they start going into these tumors, and subsequently over a period of time we can find that these tumor cells have been supplied with a fresh stock of the blood supply which contains the nutrients, food resources, as well as the oxygen for its growth and normal functioning. And what happens that as soon as these cells are provided with all the essential criteria, they start growing and developing in a much more faster rate. Now, how does a cancer is related to a cell cycle? Cell cycle exactly means that a cell has got a particular cycle after which it will die out. So a cell cycle is divided into 4 major phases, which are called as the G1 phase, M phase, G2 phase, and G1 phase, S phase, G2 phase, and the M phase. The G1 phase, where the cells start growing and start preparing itself for the synthesis of its DNA. In the S phase, what happens that the DNA synthesis or the replication start occurring. So from a single strand of the DNA, the daughter strands are formed, and subsequently the DNA gets replicated into a number of copies. In the G2 phase, the cell start preparing itself for the next phase of the cell division, which is called as the mitosis. In the mitotic cell stage, the one parent cell divide into 2 daughter cells having the equal number of chromosomes. So each of the cell has got a cell cycle. So what happens is the cell cycle in the cancer biology gets released because the cells in the cancer grows in an uncontrolled and undifferentiated fashion. So there is a massive production of these cells, and what happens is these cell cycles have been put up into a number of checkpoints. These checkpoints are the points where the cells are checked whether all the mechanisms are going correct in the cell or not. So the first checkpoint is the point which takes place between the G1 and the S phase. At this point, what happens that if the cell is not prepared to go into the synthesis or the S phase, the checkpoint doesn't allow this cell to move to that particular point. So at that particular checkpoint, the cell is returned back to go back to its G1 phase and complete all its mechanism and machinery processes so that all the enzyme, proteins, and whatever things are required can be synthesized, and subsequently now it moves to the S phase. The next checkpoint occurs between the G2 to M phase, where the cell moves to the mitotic or the division phase. At this particular phase also, all the machineries and the mechanisms are checked. If it is found that the cell is not having all the mechanisms correct, the cell goes back to the G2 phase to complete all the mechanisms and the machineries which are required to go into the M phase, and then it moves to the M phase. And finally, the third major checkpoint is in the M phase, where the mitotic or the divisions takes place. In each of these checkpoints, there is a production of cyclins, which is a protein, and subsequently these cyclins go and attach to the CDKs, which are called as the cyclin-dependent kinases. So these checkpoints are something like the border checkpoints which are present between the 2 states. So whenever we cross one state to the other state, all our vehicle documents are checked at that particular level. So if we are found that there is some missing documents, we are either fined or we have to go back to our own state to make those documents, and then only we are allowed to go to the next state. So at these border checkpoints, the same things happen. So this is same thing which is happening in the cell. So what happens is, if suppose the cell is not prepared to go into the M phase, so the cell won't move from the G2 to M phase checkpoint. So the cell will go back to its G2 phase and prepare all the mechanisms, cyclin-dependent kinases, enzymes, so that now it can have all the things ready, and now it will move to the next phase. So what happens is, all these checkpoints in the cancer cells get released. So the cell start moving from one phase to the other phase without any checkpoints or stop points. So whether or not the cell has made all the enzymes, proteins for its normal growth, it becomes irrelevant, and the cell goes to the next phase where the development and the active replications and the uncontrolled growth start taking place. Now, the cell divisions is mainly divided into two phases: the regulation of the cell division and the inhibition of cell division. So what happens in a normal cell, there are certain growth factors which are the extracellular factors. What happens is, suppose when a cell is ready to divide, these growth factors go and bind to the receptors which are present on the surface of the cell or the plasma membrane. So as soon as these growth factors go and bind to these receptors, they go and synthesize or activate certain transcription factor which goes and activates the mRNA, and the proteins which are necessary for the cell divisions are generated, and the cell is now ready to divide into its daughter cells. Now, similar thing happens in the regulation of the cell division, where the cell division inhibition occurs, in which when the cell doesn't want to divide, what happens is there are lots of growth inhibiting factors. What it does is it goes and binds to the receptor. These receptors activate certain proteins or the transcription factor, which goes and transcribe or translate the DNA or the RNA, and subsequently the proteins are synthesized which block the cell division, and the cell is unable to move into the cell division phase. So the cell is not dividing at that particular time. But what happens is, in the cancer biology, the cell loses these kind of property of inhibition, because the cell has to actively grow into number of its progeny by uncontrolled growth. So the regulation of cell division inhibition is released, and the cell actively grows over a period of time. Now, there are certain major cell types which have been found. The first major cell types is the carcinoma, which are the frequently found cancer cells, which are found in the epithelial cells which lines the organs and the surfaces, like the lung cancer, colon cancer, breast cancer, and the prostate cancer. The second major type is the sarcomas, which occurs to the fibroblast cells, mainly which are involved in the bone and the muscle cells. The third major type of the cancer is the hematopoietic cells and the organs, for example, which are involved in the red blood cells, white blood cells, which are called as the leukemias, lymphomas, myelomas. And the fourth major type of tumor is the neuroectodermal cells, which majorly affect the neuroblast cells. In this diagram, we can see the carcinoma is occurring to the tumor is occurring to the cells that cover the inner and the external body surfaces, for example, lungs, breast, colon, bladder, prostate in the case of the men. And the leukemia, lymphomas, which comes under the hematopoietic type of the cancer. The leukemia are the cancer which involves the blood cells, while the lymphomas are the one which involves the lymph nodes and the tissues. While the sarcomas are the cancer which involve the cells which are present as the supportive tissue, for example, the bone and the muscle cancers. Now, the cancer cells have got few properties which are different from the normal cells. The first property is the contact inhibition, which is present in the normal cells while it is absent in the cancer cells. The third property is the growth factor requirement property. The third property is the anchorage dependence, in which the cells are anchored to the surface to which it is bound. So it is present in the normal cell, but in the cancerous cells, this property has been released, because the cancerous cells have to move from one part of the body to the other part. The fourth major property is the cell cycle checkpoints, which are intact in the normal cells and which are absent in the cancer cells. And fifth is the karyotypic profiling, which is the number of the chromosomes, the mutations, which is absolutely normal in the case of the normal cells, while it is abnormal in the case of the cancerous cells. And the last property is the proliferative lifespan, which means for how much time the cell will divide. For normal cells, it is finite. It is known that for how much time or how much hour it will divide, after which it will go for a programmed cell death, while the cancer types becomes the proliferated rate becomes infinite and they becomes immortal, means they grow over a period of time without dying out or without getting into the programmed cell death or the apoptosis process. Over here we can see a picture which I have put showing the normal cell types as well as the cancerous cell types. If we can see the normal cell types, they are evenly spread throughout the surface. So there is a presence of contact inhibition, while there is a anchorage dependence also, means all these cells are anchored to the surface on which they are growing. While in the case of the cancer cells, we can see that there are clumps and the tumor cells which are formed in a lumps, and they have got released from the surface and the anchorage dependence property has been released, so they have become spherical or round in shape, and these clumps through the help of the blood vessels will now move from one part of the body to the other part of the cell. So we can easily see under the microscopes or the electron micrographs the cancer cells as well as the normal cells, and we can easily distinguish that how the patient is suffering. This is what is being done in the biopsy. Whenever a patient is suspected to have a cancer, the tissue part or a small part, cellular part from that particular body part is taken and it is grown in a culture medium condition, and over a period of time they are checked how the properties of these cells are being found. If there is a anchorage dependence, contact inhibitions and finite cell growth which is found, then we can say that the patient is not suffering from cancer, it is a normal case. While if the patient is suffering from a cancer, all this property will be released and abnormal cell growth and cell division will be found, and these cells will be growing over a period of time into a number of progeny, and a unregulated growth or clumps can be found in this cell culture medium. Now the cancer has been mainly found that there are certain genes which when mutated in the body causes the cancer. The genes have been divided into 4 major categories. The first one is called as the proto-oncogene. The second one is called as the tumor suppressor gene. The third one is called as microRNA gene. The fourth one is called as the mutator gene, of which the first 2 genes are the most important genes and which have been found frequently mutated in most of the cancer cell types. The proto-oncogenes are the genes which functions normally in the body cells. But what happens is whenever there is some kind of mutation which occurs in these proto-oncogenes, they get transformed into oncogenes, which is the lethal or the cancer forming part of that particular gene, and the cell divisions becomes uncontrolled in that particular cell. The second category of the genes is called as a tumor suppressor gene. As the name is suggesting, the products or the proteins generated from these genes helps in inhibiting uncontrolled or unproliferation growth. So whenever there is an uncontrolled growth, these gene products stops or inhibits the uncontrolled growth. The third is the microRNA, which functions normally to silence the expression of the genes which are not required for the growth which and when required. The fourth major category is called as the mutator gene. These genes are majorly involved in the fidelity and checking whether the replications and the other processes or the mechanisms had occurred correctly or not. So these genes are also mutated because in the cancer cells it has been found that the DNA in which the genes are mutated, the genes becomes mutated or there is some kind of chromosomal or genetic abnormalities. So if these genes are active, the proteins generated from these genes will immediately recognize that there is some kind of DNA damage, and then they will stop this cell to grow, and they will make the cell to go into the apoptosis or the programmed cell death. That's what the cancer cell doesn't want to go into. So all these genes are mutated some way or the other in the cancer cells. We will take into over here the first, the proto-oncogene, then subsequently we'll discuss some of the examples of tumor suppressor gene. Now over here I have taken an example of a colon cancer, colorectal cancer, in which these cancers, most of the cancers are a multi-step and multi-level mutations occurring in a number of genes. Over here there are lots of genes we can see have got mutated. So of these, APC, DCC, P53 are the three major tumor suppressor genes which have got mutated. So we can see in the starting there is a normal colon cells. What happens is when the APC tumor suppressor genes get released, the function get released or the gene gets mutated, there is an increased cell growth or the neoplasm start growing at that particular part, means there is a small tumors which is forming. Subsequently, there are other DNA hypomethylation and the oncogene formation of the Rous gene, which makes the cell grow into next phase of the tumor, which is called as the adenoma, which is a benign tumor. And subsequently, when the P53 gene, which is one of the most critical gene to be which has been found mutated in 50% of the cancer, when it gets mutated, what happens that these genes get released and the cells start dividing in a numerous way, and the cells get released from that particular position, and with the help of the blood vessels and the lymphatic vessels, it start moving to some other position, part of this body, and the metastatic stage start taking place. So we can see over from this example, the colon cancer example, that the cancer is not which is occurring within few days or few months. It is occurring through a period of decade of years or 10 or 15 years through a process of multiple step mutations which is occurring through the body. Now, the next important point is what exactly is a oncogenesis, means the tumor formation, and by which means or how it is caused or how the growth start occurring. Over here I have put a picture of a mammogram which is showing a lump of tumor in the breast cell. So we can easily say that the female patient is suffering from the breast cancer in which the lump is started growing. These oncogenes or the cancers are majorly caused by three major reasons. The first one is the genes which are introduced by the tumor viruses. There are certain retroviral tumor viruses. What happens is they transfer the genes, the oncogenes, into the normal functioning genes, and when these genes get incorporated into the normal functioning genes, they get transformed into the tumor cells. Through the second major process is the gene mutation, which occurs mainly in the tumor suppressor genes as well as the proto-oncogenes. The third major phenomenon through which these mutations are occurring is basically through the chemical carcinogens or the radiations which are called as the mutagens. So all these things are resulting in the cancer formation. Now, the tumor suppressor gene was first discovered way back in 1966 by the scientist Rous, of whom the name is said. The virus is called as a Rous sarcoma virus because it is affecting the sarcoma cells. So for this noble and the pioneer work, the scientist was awarded the Nobel Prize in 1966. Subsequently, there were number of these tumor viruses which were formed, of which the Rous sarcoma virus is one of the most essential part. Then there is a murine leukemia virus. Then there is avian myeloblastosis virus. So number of viruses have been found, which have been found that they transfer the oncogenic part of the human cells into the human cells, and when these oncogenic parts get incorporated into the normal functioning cells, the cells get transformed into cancerous forms, and subsequently these cells start growing in an uncontrolled fashion, and the patient starts suffering from the cancer diseases. Now, how does the mutation in the proto-oncogene affects or develops the cancer? The proto-oncogenes are the genes which in a normal conditions are functioning in a normal way for the growth and the development of the cells. So what happens is in a process of time, the proto-oncogenes gets mutated. So as soon as the proto-oncogenes get mutated to form the oncogene, which is a cancerous forming gene, there is a loss of growth control, means the cells start growing in an uncontrolled fashion. So the cell which has got the mutation in the proto-oncogene has got transformed into an oncogenic form, and the cells start dividing in an uncontrolled fashion, and the first form of the benign tumor is formed. Subsequently, the benign tumor gets released from that particular part. It moves to the blood vessels to the other parts of the body, and the malignant tumor start forming. There are different classes of proto-oncogene products which have been formed, of which the major is the PDGF, which is platelet-derived growth factors proto-oncogenes. Then there are G proteins proto-oncogenes. Then there are nuclear transcription factor proto-oncogene. So all these genes have been found to be mutated in some form or the other in different cancer cases. Now let's come into the mutation occurring in the tumor suppressor gene. The tumor suppressor gene in a normal condition will act to form certain proteins, which the proteins, what it will do is this will go and inhibit the cell division whenever there is an abnormal growth or DNA damage. So whenever there is a tumor suppressor gene which is getting mutated, when both the tumor suppressor genes present in the human cell, both the alleles gets mutated, then only the patient cell starts suffering from the loss of tumor suppressor gene, and the cells start going into a uncontrolled fashion, and there is a loss of growth control over here. So there is a loss of homozygosity over here, and the cell goes into an uncontrolled growth, and the cells start dividing into a number of progeny of itself. Now in a normal condition, a tumor suppressor gene will produce certain proteins. These proteins, what it will do is it will control the cell division in a very controlled and equilibrium way. But whenever there is a mutation in these tumor suppressor gene, the defective tumor suppressor gene protein is unable to control the cell division, and the cells start growing in an uncontrolled fashion, and the patient starts suffering from the benign or the neoplasmic cancer at that particular point. Now there are different tumor suppressor genes which have been found, which are the APC tumor suppressor gene, which is present in the colorectal cancer, ARF tumor suppressor gene, which has been found in the melanoma cancer, BRC1, which is found in the breast cancer. Then there is TP53, which have been found as a transcription factor, and the mutation of this tumor suppressor gene has been found in a number of cancers, sarcomas, lymphomas, lung cancer, kidney cancer. Then the other major type is the RB, which is causing the retinal cancer or the retinoblastoma over here, and it has been found to be causing the retinoblastoma in the child. So over here I will be taking the two examples, one of the TP53, the other one of the retinoblastoma. Now the first I will take up the P53, which the TP53 gene produces a protein which is called as P53 because its molecular weight is 53 kilodaltons, and it has been found that about 50% of the cancers are having this gene mutated. So at the very early level, if we can check that whether this particular gene within the cell have got mutated, we can easily say that there is some kind of cancerogenic formation which is occurring within the body. So what this P53 gene does is it produces certain proteins which are called as P53. So as soon as there is some DNA damage which is occurring, due to which the cell should not move into the next phase of the cell division, what happens that this P53 protein goes and activates certain genes which are called as the VAF1 genes. These VAF1 genes produces certain proteins which is called as P21 proteins. These P21 protein goes and binds to the cyclin and the cyclin dependent kinases which are responsible for the movement of the cell from the G1 to the S phase. So at the checkpoint of the G1 and the S phase, the cell is stopped at that very particular level because of the DNA damage which has occurred in the DNA of the cell. So the cell moves into the G1 phase, and once the DNA damage is complete or has been repaired, then only the cell moves into the next synthesis phase. If with the DNA damage, the cell will move into the synthesis phase, the mutation which is there present in the cell DNA will be replicated in all the daughter progeny, and the mutation will go for the next generation also. So the P53 acts as a very important tumor suppressor gene and controls the cell division at the very instant level of the checkpoints of G1 to S phase. Now, let me take up another example of tumor suppressor gene, which is called as the retinoblastoma. If you can see the picture over here, this retinoblastoma's cancer is a cancer which is occurring to the child in the eye, especially in the retina. This retinoblastoma develops during the period of birth to an age of 4 years, and it is a very common eye tumor which has been found in the children. If this tumor is found in a very early age, it can be treated and the diagnosis are there, but subsequently if the child develops this retinoblastoma cancer, the treatment becomes, and the tumor becomes uncurable. So now let me take up how exactly this retinoblastoma is occurring. The retinoblastoma genes are present, which are the genes which are a major part of the tumor suppressor gene, so they play important role in the cell cycle checkpoints. So what happens in these types of cancer, these cell cycle checkpoints are released. So there are two types of retinoblastoma. One is called as a sporadic retinoblastoma, another is called hereditary retinoblastoma. In each of these blastoma, what happens that the RB genes get mutated, and as soon as these RB genes get mutated, the normal functioning of the retinoblastoma genes get released and the abnormal proteins are formed which are unable to check the abnormal cell growth, and the patient starts suffering from the retinoblastoma and the child goes into the retinoblastoma stages. Now, there are different risk factors and the modifying agents which have been found which causes the cancer, of which one of the most lethal one is the tobacco smoking and the intake. The second major cause is the overweight or the obesity. The third is the unhealthy diet, which contains a low intake of the fruits and vegetables, which forms a major part of our normal, which should be in the normal diets. The fourth part is the lack of physical activity. Fifth is the alcohol consumption. Sixth, many of the cancer have been found, they have been transmitted because they have developed because of some kind of mutational changes which are occurring due to the sexually transmitted viruses, which is called as a HPV, human papilloma viruses. There are a number of cases in which hepatitis B viruses have also caused cancer over here. The second last major point is the urban air population, because the air which we inhale, there are lots of carcinogens present in that air. So these carcinogens over a period of time causes mutations in the genes, and subsequently the cancer develops. The last is the indoor smoke or the use of solid fuels and we inhale these smoke and these things, and subsequently over a period of time the cells get mutated. Now, the treatment of the cancer has been divided into two major steps. The first one is the diagnosis. The diagnosis step is a critical step in which the patient is dissected whether the patient is suffering from cancer or not. If the patient is suffering from cancer, which particular type of cancer is there, which particular stage of the cancer is present in the patient's body, and subsequently the patient is treated with the various treatment regimes. The next major thing is the treatment part, in which it is divided into number of therapy, surgery, radiotherapy, chemotherapy, and hormonal therapy. So we will go into each of them one by one. The first one, we go into this diagnosis. In the diagnosis, the major diagnosis is being done, or the detection is done now with the help of the PET scanner. PET scanner is a positron emission tomography scanner in which the patient is incorporated with a molecule of a glucose, which is a form of a analog of a glucose, and what this is found is how this cancer growth is occurring within whole of the body. It is detected through the scanner along with the CT scan. So what this PET scanner does is it helps us to detect what kind of stage of cancer the patient is suffering, which particular type of cancer the patient is suffering from, how much metastasis or the growth of the cancer throughout the body has occurred, and subsequently what are the treatments which can be given, and when the treatments or the patient is undergoing the chemotherapy, then the patient again goes from the PET scan so that it can be found that how the treatment is working and how the patient is responding to that particular treatment regime. Over here, if you can see, there is a picture of a PET scan in which we can see that the patient is suffering from a metastatic cancer, in which the cancer have been found in the lungs, livers, bones. So the cancer had got spread up into all the body parts. This is the image which the PET scan developed. This is a three-dimensional pictures in which we get a exact information how large the tumor is, how the growth is occurring, where exactly the tumor growths have occurred. So the PET scanner is one of the most important scanner which has become now, and most of the tumors are detected with the help of the PET and the CT scan. Now, there are different therapies in the cancer treatment regimes, which include surgery, radiation therapy, chemotherapy, hormone therapy. Depending upon the cancer type, the patient is recommended either one of these therapy or a combination of these therapy. Let's take up these therapies one by one. The first one is the tumor surgery therapy. These therapies, these surgeries are done when the cancer is majorly a benign cancer which is designated to one particular area. So the cancer or the tumor is taken out from that particular area, and the adjoining area are treated with proper drugs so that the cancer spread can be diagnosed at that particular level. The major therapy is called as the radiation therapy, when the patient has started developing the next advanced stages of the cancer, which is the metastatic cancer. This radiation therapy occurs with the help of the high energy X-ray, which kills the cancer cells. But the major disadvantage of using this radiation therapy is the normal cells also get killed along with the cancer cells. So these therapy are generally given externally or internally, and the patient has to go for a regular check when the patient is undergoing the chemotherapy drug radiation therapy. The major side effect of these radiation therapies is skin irritation, weight loss, loss of appetite, there is digestive problem, hair loss is also there, urinary problem is there, then there is fertility problems which also occurs. Of these, the major side effect is the hair loss, which is called as alopecia. What happens is during the radiation therapy, the patient suffers from the hair loss, and these hair loss occurs starts from about 2 to 3 weeks after the first treatment is done. So when the treatment gets then the treatment gets finished, subsequently the patients go back to its normal state, and it has been found that after 3 to 6 months after the treatment, the hair grows back in a proper manner. So the major side effect of these radiation therapy is the hair loss. The major therapy which is now present is in the form of the chemotherapy. In chemotherapy, various drugs are given to the patients in the form of intravenous, orally, or injection forms at the point where the cancer has occurred. These drugs include fluorouracil, doxorubicin, paclitaxel. Various forms of chemotherapy drugs have been found, and they are suggested or recommended for different kinds of cancer. So the major disadvantage of these chemotherapy drugs is they also affect the healthy cells also. So the major side effects of chemotherapy is there is a suppression of bone marrow, so the bone marrow doesn't function properly, so there is not a normal functioning or the generation of the red blood cells or the other cells which are produced from the bone marrow. Then there is a neutropenia which occurs, which means that there is a low number of white blood cells, means the patient's immune system decreases, the patient's red blood cells decreases, and the patient suffers from anemia. Then there is thrombocytopenia, which means there is a low number of platelets, means the infection chances also increases. There are gastrointestinal infections which occurs, there is an appetite loss, and the fatigueness which is present. The last therapy which is there is called as the hormonal therapy, in which the hormonal drug therapies are given to slow down the growth of the breast, prostate, or the uterine cancers, and these are usually given for a longer period of time, and this is one of the most recommended way of treatment of various cancers which are affecting the breast, prostate and the endometrium cancers. Now, it has been found that about 1,86,000 people could be saved from this cancer disease if they have got a very proper healthy diet and if they do a regular exercise. And a similar number of cases of about 1,86,000 people can be saved each year if they don't consume tobacco or inhale in the form of the cigarettes. So what we have to do at this point of time, we have to take the precautions and all the measures so that the cancer can be stopped at the very early level before growing into the next level. There are number of anti-cancer fruits which are present, which have been said, of which grapes form a major part. Then the apples are also there, citrus fruit, which are a major source of antioxidants. Then there are pomegranate, which are called as anar, strawberries are also there. So all these have been found to be anti-cancerous fruit. So if we take these fruits regularly with our normal diets, I suggest that the patient won't be having any kind of early growth or any kind of these kind of disease conditions. So with all these information, I end the lecture over here. Thank you very much.
Definitely, sir. You have raised some important issues, as well as you have raised how it can be controlled and how we can protect ourselves from diseases like cancer if we have a healthy diet. Now the question arises, does the problem of cancer is a heredity? Can it is possible?
It has been found that about 30% of the cancers are hereditary cancers. So they are heredited from the parents' lineages. So, and the cancer is not occurring because of one gene which has got mutated. So in the hereditary thing, if we have got, suppose we have got 3 mutated gene, and over a period of normal lifespan, we get another 3 or 4 mutations occurring in the different levels, tumor suppressor genes or the proto-oncogene, then we are going into a cancerous state. So in the 30% of the cancer, majorly in the breast cancer or the prostate cancer, lung and the colorectal cancer, it has been found that many of the cases are from the hereditary. So hereditary cancers are forming a major part of the development for the subsequent levels.
You have raised term mutations, but if we talk about mutation in a simple terms, how could we define mutation? What is mutation actually?
Mutation actually means, like in a cell we have got a nucleus. In the nucleus there is a DNA which is called as a deoxyribonucleic acid. So this DNA is a double-stranded DNA, and this DNA has got genes present within them. So there, in a one stretch of DNA, there may be thousands of genes which will be present, and each of these genes codes for a proteins or a protein which is present for particular parts of the, like suppose some genes are responsible for the hair color, some are responsible for the skin color. So all of these genes are responsible some way or the other for the production of proteins, which may be a part of a production system or they may be a part of the enzyme system. So in a cancer, what happens that these genes, which are majorly from the proto-oncogenes or the tumor suppressor genes, when they got mutated, means these genes have got a coding things which are present. When there is some kind of mutations occurring in the nucleotides within these particular genes, and these genes turn into an abnormal form, means they cannot synthesize the normal proteins or they cannot generate the normal proteins, then the mutation is called as the genetic mutation. So mutation is said that the mutation has occurred in that particular DNA, and now this DNA has moved into the state of a mutation. So that's what a mutation is defined as.
In case if a child in a womb of a mother and the mother at that time is suffering from cancer, are there chances that a child would be infected with the disease?
There might be chances, because if the mother suppose is suffering from leukemia or lymphomas, okay, so leukemia is the cancer which is occurring to the blood the red blood cells. And we know that the child which is growing in the mother womb is getting all the food resources through the umbilical cord and the blood supply. So if the blood supply within the mother gets distorted or there is no regular blood supply, obviously the child will suffer from the particular less of blood supply and less of the nutrients, and there may be chances that these red blood cells will also be transferred, because the genes which have occurred or which has gone will also get transferred into the child. And along with these red blood cell, if there is some kind of mutation which is present in the germinal cells or the mother germ layer cells, these germ layer cell mutation will also be transferred to the child. So along with the germinal cell transfer of the cancer cells, there will be transfer of the red blood cells which are suffering from the leukemia or lymphoma conditions, and the child, the newly born child which is developing inside the fetus, which is developing inside the mother's womb, also will get affected from that particular cancer development.
If we talk about tumors, as you said in the lecture also, do it is believed that yes, these are the tumors or the infections at the initial stages which are not taken care of grow into a cancer?
Yeah, because most of the cancers, what we have found is there is a early development of a small tumor which grows in a outgrowth, because many a times we found that in our bodies after 10 or 15 years there is a small outgrowth. So what we should do is, if there is an outgrow and many of the tumors are benign tumors which may stop at that particular level, but many of the benign tumors, if there is some kind of mutation which occurs subsequently, what this benign tumor goes is, it goes into the next phase, which is the metastatic phase. So what happens is, if we can detect within our body externally that there is some kind of outgrowth or a growth of a tumor which we suspect that is present, we should immediately consult the doctor that this particular growth is being done. This is generally recommended in case of the breast cancer, for the early detection of the breast cancer, because the women are recommended that they should regularly have a checkup in which if they suspect that there is some kind of tumor which is developing, they should immediately report to the doctors so that the doctors can check and go for a scanning, PET scan or the other scan or mammograph, so that they can say that yes, this is a cancerous form which is developing, and if we can detect in the initial stage 1 level of these things, we can easily go for a surgery, take out these tumor cells at the initial level, and the patient will not be suffering from any of these disease condition, and the patient will be free from all the cancer condition states. And there are a number of cases that if the cancer has been detected at the very early level, then the patient is not suffering from the cancer condition and the patient is living a normal life. Maybe he is living a life of about 80 to 90 years also. So it all depends upon how early we detect the cancer, how healthy diet we have after the detection, how we maintain our protocol and the lifespan after the detection, and we should have a regular body checkup or the regular criteria checkups of all the body vital organs. Whether we are suffering from any disease or a normal condition, we should regularly go for a regular checkups of the body vital organs.
So if we talk about oral cancer, we have seen many people have oral diseases, like they got infections from time to time, and some people are susceptible to such kind of diseases. Do they have chances in future to have a disease like cancer?
Yeah. So oral cancer has been found that many infections, sexually transmitted infections which get transferred through the oral, they also transform the cell or mutate the cell to a cancerous form. And it has been found that the cancer is not occurring because of the acute infection, it is occurring because of the chronic infection. Chronic infection means the infection which is occurring for a longer period of time. Means suppose we have got some kind of infection in our body, like suppose we have got ulcers in our liver cells or the gastrointestinal tract. If this ulcer is a chronic infection, it has been found that about in 50% of the cases, the chronic infection of ulcers have led the patient to go into a liver cirrhosis or a liver cancer. So if the infection is going for a longer period of time, then that infection is really alarming. So the patient should immediately go to the doctor and get the treatment done, because the chronic infection over a period of time causes some kind of mutation subsequently, slowly in the genes, which get transformed into a cancerous gene with the changes in the proto-oncogene, tumor suppressor gene, mutated gene, or the microRNA gene. But the acute infections are generally not found to be relevant with the cancers because they are acute. So the patients get the acute infection, he goes to the doctor, means they are in the maximum disease condition, he gets all the drugs and the therapies, and the disease conditions gets evaded out from his body. So the chronic infection plays a major role in the development of the cancer, and they have to be taken care of at the right point and the right level of the cancer detections.
Definitely. So sometimes it is also heard that cancer has affected some part of the body, for example liver, but finally after some stages doctors detect that the cancer or the infection has reached to all other parts of the body. Are those cells which have reached to different parts of the body, they have become active?
So what happens is, as you asked, what happens is, suppose there is a cancer which has occurred in the liver. So the liver cells have started growing in an uncontrolled way. So as soon as the liver cells start growing in an uncontrolled way, subsequently from the benign stage, which is the tumor growth stage, they move into a malignant stage, where the cells get released from the liver, and through the blood vessels and the lymphatic vessels, they move to some other part of the body, for instance to the lungs or kidney over here. So the liver cancer cells now have grown in the liver. Through the blood vessels, they have gone to the kidney cells or the rectal cells. Now they start growing in the kidney cells over there. Over there also they start developing in a uncontrolled or a unregulated fashion. So the patient, when he or she goes for a PET scan, which is a positive emission scan, what the doctor finds that there is a detection of the tumor present as well as in the liver, and there is some kind of scanning detection which is found in the kidneys also. So these kind of stages are called as a metastatic cancer, in which the cancer which has grown at one point of time has now gone into the other part of the vital organ, and the patients start suffering from multi-organ failure, because subsequently the cancer cell start developing to all the parts of the body, be it lung, the esophageal, rectal, colon, brain, eyes, everywhere, and the patient suffers from a multi-organ failure, and the patient dies of this multi-organ cancer.
Definitely, sir. With this lecture, we would only say that let's fight against cancer. Let's have a rich and healthy life by eating healthy diet, by having a healthy diet, regular exercises, and all what you recommended. We should, of course, incorporate in our life. As you said, we should go for yogas and exercise regularly, and this is the only mode through which we can fight against cancer. Thank you, sir. Thank you so very much for delivering such a nice lecture and giving deep insight into this very topic. And I would like to tell students that Dr. Amit Bhattacharya would be again with us tomorrow, and we would be discussing a new topic, and hope that you will be benefiting from tomorrow's topic also. Thank you, sir. Thank you so very much.